This video starts with a discussion of biosynthesis and specifically how nucleic acids are made in viruses, and this is really critical to understanding a lot of the details about viral replication. But what I've found is that if I require people to learn this, it takes so much of their attention, they don't learn the other things. So I've made these details about nucleic acids optional. So in this video I will go through the whole thing, and if you're curious, I'll take you through everything you need to know to understand the difference between a positive sense or negative sense RNA virus or a retrovirus, and how the genome type determines where in a cell a virus replicates and things like that. But you can skip this if it is labeled as CUR in the slides you've been given, like in this one. So make sure you're looking at your slides and your study guides so that you know what you're supposed to learn, because that may change from one term to the next one. So with that, let's get into biosynthesis. Okay, so before I explain biosynthesis in more detail, we need to review the way nucleic acids work. So I know you know about transcription and translation, and you know about our DNA replication, but I just want to look at a basic level at what does it take to make a copy of a nucleic acid. We're going to use base pairing. So I want to just think about DNA first. So this is going to be DNA. And we've got a 5' end, and let's put in some bases, A, C, C, G, T. Okay, that's some random bases. And then we're going to have a start codon, A, T, G. And then we're going to have codons, A, whoops, T, C, G, whatever. And then 3' end at the other end. So that's a strand of DNA. What if I want to make a copy of this strand of DNA? What do I have to do? What do enzymes in my cells know how to do? There's no enzyme in my body that can look at this and say, oh, that's an A, let's put an A here, and let's put a 5' here, an A, and then a C, and that's not how it works. Every time we copy a nucleic acid, we use base pairing. And every nucleic acid has a 5' to 3' end, and base pairing works in an anti-parallel way from 5' to 3'. So if I want to make a copy of this DNA, I have to build another DNA molecule that is what we call the reverse complement of this. How would we make this strand of DNA? How would we make this complementary or reverse complementary strand of DNA? Well, the way our enzymes actually work, DNA polymerase, RNA polymerase, any nucleic acid enzyme, it does it by base pairing. It's going to start at the 5' end, and it's going to try different nucleotides. These are floating around in the cytoplasm, and it's going to grab one and try it. It's going to try an A and say, oh, does that fit? No, it doesn't fit. It doesn't base pair with a guanine. It's going to try a whole bunch of different nucleotides until it tries cytosine, and cytosine base pairs with guanine. So it's going to put that one in the chain. It's going to do the same thing here. It's going to keep trying until it finds the one that base pairs. That is guanine. It's going to try this one until it finds adenine. And this is going to get tedious, but let's just do it. You can tell me a joke in your head. And we make the new strand. So this new strand doesn't encode the same protein. It doesn't have the start codon. It has the reverse complement of the start codon. And there's no ribosome is going to be able to interpret the reverse complement of anything. So this would be the coding strand, this would be the non-coding strand. But remember, the point was we want to copy this one. So the first step is making this strand. And then the second step would be separating them and doing the same thing again, maybe using, I don't know, plum. So we're going to do that whole process one more time. In the exact same way, we're going to try all the different nucleotides until we find the one that base pairs with this, and that's going to be adenine. Then we're going to try what base pairs with that. It's going to be cytosine, cytosine, guanine, and we extend it this way from 5' to 3'. And if I did this correctly, this sequence will match this sequence. Now that's the basis of DNA replication. That's the basis of transcription. This happens during transcription also. If this were transcription, all of these thymines would be uracils, so the T's would be U's if it was transcription. Well, this is really important for understanding viral biosynthesis, as you're going to see. We just have to think about this. And in general, what did we see? Well, we saw a coding strand, so we're going to call that a positive strand. And to make a copy of it, we had to make, a non-coding copy. We had to make that strand that is the non-coding strand. And then from that, we could make a coding copy or coding strand. We're going to see the same thing in RNA. The RNA viruses are going to need to do this. If they want to, if this is what they have, if they have a sense or coding strand and they want to make copies, they have to first make non-coding copies and then make coding copies of those. So go through this as much as you need to understand it, because you're going to need to get this to follow what's happening inside the viruses when we look at biosynthesis. So let's do that. No, really. Okay. So this is some review that's trying to remind you of what I just showed you. The DNA is in the nucleus. DNA gets transcribed using a process like what I just showed you, using base pairing to make an anti-parallel strand. The messenger RNA goes out to the cytoplasm where it's translated. And so I'm going to be using a lot of things that look like this. DNA arrow RNA, RNA arrow protein. So transcription, translation. If DNA went to DNA, that would be DNA replication. This is what I just drew. All of this replication happens via base pairing and making the opposite strand first. Then we can make another copy of the first strand. So for a DNA virus, it can send its DNA, its nucleic acid, into the host cell nucleus, where the host normally does all of this. The host will take that viral DNA and transcribe it to make viral RNA. And then that viral RNA will be pushed out of the nucleus to the cytoplasm, where it will be translated to be viral protein. Okay, that's the natural way we handle all this in our cells. The only difference is now we're doing it with viral DNA and viral genes and making viral proteins. So that's what a simplified DNA virus could do. The simplest case of a DNA virus is one that just pretends it's host DNA. And we're going to use some terminology for short. That stands for viral DNA, viral RNA, viral protein. So this was a simplified DNA virus, and I just say simplified because there are examples out there that are DNA viruses that don't look like this at all because they are extremely convoluted for reasons no one knows. The next one I want you to think about is a positive or coding sense single-stranded RNA virus. So the virion contains a positive sense RNA. And that positive-sense RNA is messenger RNA. There's no need to transcribe it. Nothing has to happen. As long as it has the signals on it for initiating translation, and as long as it's got a 5' cap and things like that, it'll be translated using the host machinery. The problem that this runs into is that the host does not have an enzyme that can make copies of this. So to make a copy of this, we would have to make an anti-parallel negative-sense RNA. The complementary strand of this we call negative-sense. And then we would use that to make copies of this. But the host cell, like our own cells, have no enzymes that can do that. We don't have enzymes that can make RNA copies of RNA molecules. We just don't. These enzymes that we don't have are called RNA-dependent RNA polymerases. So that means they have an RNA template, so they're RNA-dependent, and they make an RNA product, so they're RNA polymerases. And we don't have one of these in our cells, so one of the viral proteins has to be an RNA-dependent RNA polymerase to allow these copies to be made. And this protein, this RNA-dependent RNA polymerase, as soon as the virion is made, it doesn't need that anymore, so it can leave that enzyme behind in the host cell. So where things get complicated are for negative-sense single-stranded RNA viruses, because their genomes are not messenger RNA. They are the reverse complement of messenger RNA. So they can't be translated. They don't mean anything to a ribosome. So before any biosynthesis can happen, they need to somehow make a positive-sense RNA copy. And in other words, they have to do what I was just drawing out for you. They have to take one strand and make a complementary strand. And that complementary strand is what will be translated. So they have to make that strand by, experimentally trying different ribonucleotides, building that strand, and that will act as a messenger RNA that can be translated. And so let's think about that. If we back up, that all has to happen before any biosynthesis happens. So somehow this virus needs access to an RNA-dependent RNA polymerase at the very beginning, as soon as the virion uncoats. As soon as uncoating happens, it needs to already have an RNA-dependent RNA polymerase, because it can't rely on something it encodes to be made by the host cell. The host cell can't do anything with this. So it has to bring its own RNA polymerase in its virion. So negative-sense RNA viruses have to have an RNA-dependent RNA polymerase in the virion. Take as much time with this as you need to, because to follow biosynthesis of influenza, you're going to have to kind of get what's going on when I talk about RNA-dependent RNA polymerase and when I talk about negative sense versus positive sense. Again, just in case this was, well, I just want to say it one more time in a different way. This positive sense RNA virus, its genome is already messenger RNA. We know it's going to need an RNA-dependent RNA polymerase that's encoded in its genome, and the host is going to make that. So this process, this translation that happens right away, well, one of the products of that is going to be an RNA-dependent RNA polymerase that can make the new copies of the genome. But for a negative-sense RNA virus, that can't happen because the genome is not messenger RNA, so no translation is going to happen at first. The first thing that has to happen is it needs to use the RNA-dependent RNA polymerase it brought in the virion to make a positive sense RNA copy. So it needs to do this first, and then the cell can start making proteins based on this positive sense viral RNA strand. Okay? And then the last one that I want to show you in terms of biosynthesis is a retrovirus. And retroviruses bring up a whole different idea because they use a form of latency as an essential part of what they do. They want to insert themselves into the host genome. So why do they want to insert themselves into the host genome? Well, remember what I said a virus is. A virus is information. It is the nucleic acid sequence that encodes all the instructions for making a virion. And so if they can put that nucleic acid sequence in our DNA, they don't have to do anything else. When our cells divide, we are going to make a new copy of their viral genome. And that is what a lot of viruses want to do. They want to wait for better conditions, and so they want to put their information in our genome. Our genomes are packed with viral information. There are, I think, hundreds of viral sequences in a typical person's DNA. Some of them are ancient, and they're not functional. So we evolved in a way that mutated and broke those sequences. So they're waiting for us to make new copies of them, and we never will. But latency is a functional thing where they can hide in our cells. So a retrovirus has a positive sense RNA genome, and the virion has to contain an enzyme that's going to change this as soon as it gets into the cell. The enzyme it has to have is a reverse transcriptase, that is an RNA-dependent DNA polymerase. So it uses an RNA template to make DNA, and that's backwards. 40 years ago, no one thought that could possibly exist. So reverse transcriptases do this. They take the viral genome and they make DNA copies of it. But remember what I showed you here. Let's say that this was reverse transcriptase, and this were the RNA sequence in the genome. They'd make this DNA strand, and then they'd need to make another DNA strand to make double-stranded DNA. DNA has to be double-stranded. And it turns out that reverse transcriptase also has to be able to do that second step. It needs to be able to do DNA replication. So reverse transcriptase does these 3 steps. It goes from a positive sense single-stranded RNA to a negative sense single-stranded DNA. And then it makes a positive sense single-stranded DNA. And that allows the DNA to be double-stranded. These would base pair with each other. And then it uses, from there, it can make viral proteins that stick it in our own DNA. And then it hides and it waits. It can wait forever or until the time is right to start making virions. So we call it a provirus if it is in our genome but not making virions. And eventually the host will start transcribing the genes and finish the replication cycle, but that could be years later. And so while we're talking about latency, I just want to finish that topic. A lot of viruses do that. The simplest way is to incorporate their genome into our genome, to hide as our own DNA. They just put their information in our genome. They use genetic recombination to put their DNA sequence into our DNA. Retroviruses do this. A lot of bacteriophage do this. But there are other types of latency. So I was just reading about one of the herpes viruses that hides in the trigeminal ganglia. It hides in neurons in the trigeminal ganglion, and it prevents any of them from going through apoptosis. So they become truly immortal, and that means it can hide in that one cell for your entire life. So if you think about, I don't remember which one this was, but if you think about the varicella zoster virus that causes chickenpox, if you get chickenpox, then this virus goes and hides in your neurons until you're 65 years old, and then it goes and causes shingles. So it's literally hiding in the same neuron for decades. It's kind of amazing. Okay, well that is plenty for this video on viral replication. It's the more you think about the level of detail I've put in here, the more you think about it, the more sense it will make, and you'll be able to make sense of the example that's coming up. So spend the time with it, ask me questions. I'm happy to work through this with you in office hours. So I'll see you at the next video.